be delivered by Pierre Cardol.
Yeah, I'm here.
Okay.
Can you see my screen?
Yes.
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 Okay, thank you, Anna. So my name is Pierre Cardol. I'm working at the University of Liege, Belgium. My main interest is bioenergetics of mitochondrion and chloroplast and their potential interaction. So I started working on that since my PhD thesis, mainly on green cellular algal models like Chlamydomonas reinhardti. And then less than 10 years ago, I moved to more to complex algae, let's say, like euglenas, dinoflagellates, or diatoms. So the results I'm going to show you and summarize very briefly are part of a work that has been funded by the European Research Council from 2016. First thing maybe of interest for the world community is that we've been working with my colleague Denise Borin in Liege on the transcriptomic data. So we generated some raw transcriptomic data from excuse me. There. Yes, my girl is entering just right now in the room. That's the funny part. So we generated our own RNA-seq data, and we also have been working on data available in the literature and generated by colleagues. So we reassembled individually the raw reads. So the total reads is 2.6 billion of reads. And so we get in independent assembly, we cleaned as much as we can from residual contaminant. And then we merge these 5 transcriptomes to generate a consolidated transcriptome, which now totalize more than 90,000 transcripts for about 50,000 predicted proteins. So this work has been submitted. Then from that, we were wondering if it was possible from this comparative transcriptomic data set to use a clustering approach to study co-expression between some genes. So for that, we studied the most variably expressed transcript. We selected 2,500 transcripts and generated 36 co-expression clusters. Unfortunately, what we found is that from those data sets, the results are mainly driven by the study, so by the lab, than by the experimental condition. It means that despite the fact that most of the labs studied light, dark, acetate, and without acetate, there are more difference between the laboratories than between the experimental conditions. To come back on bioenergetics, so one thing that we studied is the subunit composition of the mitochondrial respiratory complexes. And what we found is that each of the complexes that transfer electrons from NADH to oxygen, so complex 1, complex 3, complex 4, as well as the mitochondrial ATP synthase, comprise a mixture of canonical subunits, so subunits which are found in model organisms like yeast, mammals, land plants, and some subunits which are exclusively shared by trypanosomes. So the next steps in the lab was to get some insight into the structure of the respiratory complexes. And what we found is that each of those complexes, and you can see that following the red arrows, presents extra structural domains, which are probably composed by these additional subunits which are found in trypanosomes and euglenas. So that's quite interesting, but that's all I can say up to now, because we don't know yet the function of those particular subunits and extra domains. So this work has been published during the last couple of years. So the next part of the work was to study by the same approach, the subunit composition and structure of the photosynthetic complexes. And I only show you during this presentation the results about Photosystem II. So the picture on the bottom left is an overlay between the crystal structure of land plant Photosystem II and the picture that we obtained by electron microscopy for PS2. So there are some important differences, and one of them is a missing density in Euglena, which corresponds to a very well conserved antenna, which is the antenna CP26. And in purple, an extra density, which corresponds to an antenna CP24 in land plants, which is absent from Euglena. And we think that this extra density might correspond to a special LHC antenna, which has been described already a long time ago, and which possesses a very unusual absorption and fluorescence emission spectra, and comprises only chlorophyll A and diadinoxanthine, so 2 pigments. With respect to Photosystem II, Photosystem II is one of the major sites for photoprotection of the photosynthetic machinery. And as most of you know, Euglena is more sensitive to highlight than other photosynthetic flagellates. So this is illustrated on the picture where we compared a different light regime from low light to the left part to high light to the right part. Chlamydomonas reynardi on the left and Euglena glacialis on the right. So Euglena is light sensitive, and we found that it's associated to a lack of most of photoprotective mechanisms that have been described in model green plants. So meaning there is no xanthophyll deepoxidation, there is a lack of classical antenna, which have been found in model green plants. And overall, this is associated to a lack of thermal dissipation. And final slide is to show you our last results, which are also not yet published, where we describe a strong interplay between mitochondrion and chloroplast. And we have strong evidence to conclude that X-ray and ADPH, which is produced in the chloroplast, might be exported and imported into the mitochondrion, where it's used to generate ATP, which is then re-imported into the chloroplast. And together, NADPH and ATP participate to CO2 fixation through the Calvin cycle. So just to picture, one to show you that when grown under different conditions, there is a strong positive relationship between photosynthesis and respiration in Euglena. And electron microscopic picture of wall cells usually show, let's say, a co-localization inside the cell between mitochondrial in red and chloroplastic in green sections. Well, this work is not yet published. With that, I'm done, and I thank you for your attention. I have to just to add one thing. If you have specific question about the assembly and the consolidated transcriptome, you should directly contact Denise Borin, which is attending to the meeting right now. Thank you.
Thank you, Pierre. And now we'll